I'm Dr. Philip Toleikis, President and CEO of Sernova Corp. Sernova is a publicly traded company listed on the Toronto Venture Exchange and also on the OTCQB in the United States. Please see the forward-looking statements. Sernova is a regenerative medicine company. With respect to regenerative medicine, our focus is at the cell therapy level, where we are looking at providing a functional cure to treat chronic diseases such as type 1 diabetes and a number of other indications. Sernova has taken a stepwise development approach and has led to significant clinical success, and I believe we're the first company in the world to show clinical success in this field. With respect to being an innovative leader, Sernova has an integrated therapeutic solution that consists of an implantable device which forms an organ-like environment for the placement of therapeutic cells. And we then can protect ourselves from immune system attack within the device itself. So the cell pouch overcomes a number of different barriers with cell therapeutic solutions that have been shown previously, and I will go into some details around this. Sernova has a phase 1/2 active clinical study for type 1 diabetes, and we have already shown that our cells are able to produce insulin into the bloodstream, and we've shown very positive clinical efficacy indicators for patients with type 1 diabetes. We also have proof of concept studies that have been completed with human cells for hemophilia A and also thyroid disease. So with respect to our platform approach, there's 3 parts of our technology. The first is what we call the cell pouch. This is an implantable, scalable medical device that goes under the skin, and it provides an organ-like environment for the placement of therapeutic cells. And we begin our clinical trials with human donor cells because those are the real thing that we know work in your body, but we also have stem cell-derived technologies for diabetes and other areas. And we are also working with major pharmaceutical companies with collaborations for the potential of licensing these therapies to our company. We also protect ourselves from immune system attack, either through medications, and we also have brought in several local immune protection technologies called conformal coating. And we're also working on a collaboration with a company called Ajax with respect to using the HLA gene to protect cells from immune system attack within our stem cell-derived technologies. So our cell pouch is manufactured in the US under GMP conditions and meets regulatory standards for US, Canada, and Europe. We have 2-year real-time stability on our product, and we can manufacture as many devices as we require. And our approach is that we're looking at treating millions of patients going forward, and so we are set up to be able to produce the pouches to be able to do that. From the patent protection side of things, we have worldwide patent portfolio around composition and use of medical devices and delivery of cells and applications, glucose responsive stem cell derived technologies, and local immune protection technologies. And our coverage is full patents and claims around the world, including North America, South America, Europe, and Asia. With respect to the device itself, there have been a number of issues with a number of different kinds of devices placed in the body for therapeutic cells, and Sernova, we believe, has solved virtually all of these issues, including fibrosis, scalability, vascularization, et cetera, and I'll talk about that as we're moving forward. And our device have been proven to work with human cells in patients with stem cell-derived technologies and also with tissues. So the reason that our device and our therapeutic cells are working is because we believe we have a biologically compatible delivery process. So what we do is implant our device under the skin in a very rapid procedure. And after several weeks, the device has filled in with highly vascularized tissue to form these beautiful tissue chambers for the placement of the therapeutic cells. So we'll go in and place the therapeutic cells into the device chambers, whereby they have an immediate environment to be able to survive and function. So these cells are sitting in a vascularized tissue matrix. And then that allows them to not only survive, but able to produce their proteins or hormones and release those into the bloodstream as required. So our first clinical application is type 1 diabetes. And I think everyone knows that there are over 400 million people worldwide with this disease, and about 10% of these have type 1 diabetes. And what we're really doing is looking for a functional cure for these patients. And that means that what we're doing is replacing the cells that have been lost in the pancreas in this organ-like environment so that the patients will not have to take insulin injections anymore. So this is has the potential to be the biggest therapeutic advancements for type 1 diabetic patients since the discovery of insulin 100 years ago. So with respect to our therapeutic pipeline, our first clinical applications is using human donor cells, and we are currently conducting a phase 2 clinical trial at the University of Chicago, and I'll be presenting some important data that we have released in that study so far. Our next program involves locally immune protected stem cell derived technologies that we will also be moving into the clinic for type 1 diabetes. In addition, we have a program in hemophilia A where we have already shown that patient-corrected cells for the factor VIII gene can be placed into our cell pouch in preclinical models and recover the function of the bleeding disorder that has occurred. And then from the thyroid perspective, we are the first company in the world to be able to be working with patients that are having their thyroid glands removed. And we've been able to show that we can take thyroid tissue and place it into cell pouch and have that tissue survive and provide precursor thyroid hormones into the bloodstream in preclinical models. So with respect to our lead diabetes indications, we're focusing on patients that are the most severe patients with this disease. And these patients have what we call hypoglycemia unawareness. And this affects about 15% of patients with type 1 diabetes. And basically what happens in these patients is that when their blood sugar levels start to drop, they're due to an insulin injection, they're unaware of that deep drop of blood sugar, which could result in the patients going into a coma and without even realizing it. So these are the patients that you hear about in the newspaper, especially children that will not wake up in the morning simply due to the fact that they have had this hypoglycemia unaware event that they're not aware of. So we have already conducted a first in human trial, and we have shown that the device is safe in the body and that the cells within the device are surviving. They become highly vascularized and they're able to produce insulin. So this was a very good first step for our device and our therapeutic cells. We have now embarked on a phase 2 clinical trial at the University of Chicago. And with respect to the design of this study, it is a safety efficacy study. It's an open label, company sponsored IND. Primary objective is safety. Secondary objectives are to review key efficacy indicators that are important for patients with type 1 diabetes. The first is survival of endocrine tissue in the cell pouch. And here we remove a sentinel device at 90 days and have it independently assessed by pathologists to ensure that the cells are surviving. Then next, we're looking at the proportion of patients with a reduction of severe hypoglycemic events, HbA1c reduction, which is a long-term indicator of diabetes. And we're also looking at 20 different additional endpoint analyses. So with respect to some efficacy data, our clinical investigator has presented data at a number of different conferences. And basically what we have seen in our most advanced patient is a significant reduction in hemoglobin A1c over time with several doses of the cells in the device. We have also shown a significant reduction in long-acting use of insulin, all the way down to no need for insulin use, and also a reduction of short-acting insulin. And importantly, we've also shown a complete elimination of severe hypoglycemic unaware events. The other important factor here is that we know that our cells are producing insulin because we measure that in the bloodstream by a biomarker called C-peptide that is only released when insulin is being produced by therapeutic cells. The other part of our study is that we're doing continuous glucose monitoring in our patients. And on the left-hand side, you can see in the green area is where you want your blood sugar levels to be able to be stable in. And we see that this patient is dropping below that area, going into the hypo events multiple times a week. And we see that after a number of cell transplants, we're finding that the patient actually completely eliminates the hypoglycemic events. And you can see on the right-hand side that the tracing is much more stable from that perspective. We have also shown with respect to our Sentinel device that the islets within the device are surviving, that they are producing insulin, and they're highly vascularized. And this has been independently assessed by a pathologist that reviewed these results. So with respect to our phase 1/2 safety work, we have shown that the cell pouch is always well incorporated with vascularized tissue and is shown to be safe in the patients. And we're showing no incidences of severe adverse events that are related to the cell pouch. We have also been, the study's been reviewed by our Data Safety Monitoring Board, which is independent in both 2020 and 2021. They gave it a clear sail, indicating that that they recommended the study continue as designed. With respect to ongoing safety and efficacy in our patient population, this is a 7-patient study. We've enrolled 5 patients, and they have been implanted with cell pouches and ongoing transplants. And we're showing positive safety, and it's well tolerated in our ongoing patients. And with respect to efficacy, Sernova is the first regenerative medicine company to demonstrate with a subcutaneous transplanted device that we are achieving persistent islet graft function in our diabetic patients. And we're also showing a number of features such as sustained levels of C-peptide, which proves that we're getting insulin to the bloodstream, reduction of HbA1c long term, and an improvement of continuous glucose monitoring, including reduction of hypoglycemia unaware events. And very importantly, we're showing a reduction in the need for exogenous insulin, even down to a patient that has been off of insulin for over 10 months now. The other important thing is that our investigator has reported that a reduced dose of islets actually improves the function of the cells within the device itself. So this is going to be very important for stem cell derived technologies as we're moving forward. So with respect to the diabetes opportunity, we start off with our smaller population, but we believe we can get that approved really quickly in a number of years, short term with revenues coming in. And then as we're moving forward with our stem cell derived technologies, then the market goes into the multi-billion-dollar applications. And with respect to competitive landscape, we believe that Sernova is the only company in the world that has absolutely proven that our device is functional, the cells are surviving, and that we're getting clinical efficacy data from that perspective. With respect to clinical summary and progress, we have been working on this for a number of years, and in 2021, we're continuing to bring data in, and we're going to be completing enrollment in the study. With respect to our hemophilia A program, this is an established program that has gone through preclinical studies and will be moving into the clinic over a period of time. And our thyroid disease program is very, very similar. We've already conducted preclinical studies, and this program is something where there are approximately 150,000 patients who are having thyroidectomies in North America alone that we will be able to treat. So with respect to our action plan going forward, we're going to be moving our local immune protection technologies towards the clinic. We're going to be completing the enrollment of our phase 2 study. And from a hemophilia A perspective, we're moving on with, and also our thyroid program, we're moving on towards preparing for clinical trials in those areas. Our management team has had over 20 years experience on an individual basis with expertise in the cell therapy application, medical device application, and immunology. So I'd like to stop there, and thank you much for the opportunity to present.